Right, today we are going to talk about bacteria, and all of you know that it's very important to classify bacteria as if they are obligate anaerobes or they are aerotolerant anaerobes or they are facultative anaerobe or microaerophilics or obligate aerobes. It's very important to classify bacteria according to that because these five categories, they behave differently in the pathogenesis of infections and diseases and also their treatment by the drugs, right, or by the pharmacological agents is different for anaerobes and different for aerobes. Now, before we classify bacteria into these subclasses, the very important thing is we should know the relationship of bacteria with oxygen, right? This classification is based on how bacteria deal with oxygen. Now, there are two aspects related with bacteria dealing with oxygen. Number one, that do bacteria use oxygen in energy production or they do not use oxygen in energy metabolism. This is one thing, right? When we are classifying bacteria in such way, we must be very clear that what is the relationship of bacteria with oxygen in energy metabolism, in production of energy or ATP, right? Secondly, second aspect which is also extremely important, that we must know what is the relationship of bacteria with oxygen related to their self defenses against the reactive oxygen species. As you know that wherever oxygen is present, right, it can react with different biologically active molecules, right? It may be our cells, animal cells or plant cells or even microbe cells. When these cells are exposed to the oxygen, right, different biochemical reactions which I will teach you later in this lecture, those biochemical reactions produce highly reactive, unstable, dangerous oxygen derived radicals or reactive molecules. As a group, those molecules are called reactive oxygen species, right? Because reactive oxygen species are produced in any biological system which comes in touch with oxygen to survive in oxygenated environment and to multiply in oxygenated environment, the biological system must know how to deal with reactive oxygen species, how to detoxify reactive oxygen species. So whole this lecture will revolve around initially about two basic concepts, that energy metabolism and role of oxygen in bacteria, and secondly, what are the bacteria having type of defenses against the reactive oxygen species, and then eventually we will, in the end, we will classify bacteria according to the upper classification. Let's come back and deal with first thing, right? That what is the role of oxygen in different type of bacteria energy metabolism. We say basically that there is anaerobic energy production, right, and there is aerobic energy production. When we say anaerobic energy metabolism, we mean that basically when nutrients are broken down, when nutrients are catabolized or broken down to release energy and capture energy, right, oxygen is used or not as final electron acceptor, right? Let me briefly tell you. When, let's suppose we have glucose, right? If a bacteria gets glucose, now to extract energy out of glucose, bacterial metabolic pathway has to break down the glucose into its components, right? And energy should be released in the form of electrons, and those electrons in the end should be accepted by final electron acceptor. If final electron acceptor in energy metabolism is oxygen, we say there is aerobic respiration, right? But if final electron acceptor is not oxygen, then we say the energy producing mechanism is not aerobic, it is anaerobic. It is not aerobic, it is anaerobic. Now, billions of the years back, right, about 3 billion years back, right, when life started and bacteria were there originating during the evolution, right, initially Earth was not having enough oxygen. Rather, in the very beginning, in a very ancient times, there was no oxygen in the atmosphere. There was no oxygen in the atmosphere. So the life forms which were evolving at that time, they have to evolve and develop such mechanisms to develop energy which are independent of oxygen, right? Then a very big event occurred, that we call it big oxidation event in the evolution history. What was that event? A special type of bacteria came into existence. We call them cyanobacterium. Now cyanobacteria were able to produce oxygen, and over the millions of years they became so abundant that they produce huge amount of oxygen, and now our atmosphere has about 21% oxygen. Is that right? So basic thing is that when life evolved initially, it evolved in the world where there was no free oxygen available. So it means energy metabolism had to run without oxygen. So they had to be anaerobic energy metabolism. They had to be anaerobic energy metabolism. But as organism evolved and cyanobacteria made lot of oxygen, right, then these primitive bacteria which were originally anaerobic metabolism running, these bacteria had to evolve further, right? Arrival of oxygen had one good news and one very bad news. The good news was that if life forms could evolve in such a way that they could use oxygen in the energy metabolism as final electron acceptor, then lot of ATP could be made, lot of energy could be made, right? This was the advantage of using oxygen in energy metabolism. It means the very big advantage was that if bacteria could evolve in such a way that once oxygen is available, right, they should convert their anaerobic metabolism into aerobic metabolism. And advantage is they will produce lot of energy, because anaerobic processes produce less energy, less ATP, and aerobic processes produce more energy, right? And if there's more energy, there's more ATP, then of course they could survive better, they could multiply better, and they could grow better. So this was a big advantage. But oxygen also brought a very big disadvantage. You know, every good thing has a dark side. These days, the way we learn medical sciences, we think oxygen is always good. But this is not true. Oxygen is good because when it is used in aerobic respiration, it gives a lot of energy. So far, so good. But there is one trouble. The trouble is that exposure of metabolic pathways and biological system to oxygen produces lot of reactive oxygen species, which I will explain later in this lecture. Those reactive oxygen species or reactive oxygen related molecules, they react, they are very unstable and they are very reactive, and they damage DNA, they damage RNA, they damage proteins, they damage lipids, they destroy membranes, and life is not compatible. Now we are in trouble. What is the trouble? That when living organisms, microbes or plants or other or even animals, when their cells are exposed to the oxygen, right? Even though there is a big advantage that oxygen can help in producing a lot of ATP, but there is a disadvantage that oxygen can also produce reactive oxygen species and that can destroy the cells. So what happened? During the evolution, all organisms were forced and selected in such a way that they should develop defenses against oxygen derived reactive radicals or molecules. They should have defenses against what? They should have defenses against reactive oxygen species. And for those defenses, many mechanisms evolved. Some of the mechanisms were, they developed special enzymes, which I will explain later, like dismutases, catalases, peroxidases. These enzymes could detoxify reactive oxygen species. So in this way, when our biological systems or microbes, they evolved into such cells which were producing dismutases, they were producing catalases, they were producing peroxidases, then they were able to neutralize or detoxify reactive oxygen species, and then they could survive. So what is the whole discussion up to this moment? Up to this moment I have only told you one thing, that anaerobic organism, anaerobic energy metabolism is a primitive metabolism, less energy, right? Once the oxygen appear into atmosphere, oxygen, new metabolic pathways develop to take the advantage of the presence of oxygen, and those mechanisms were more efficient, and oxygen dependent energy metabolism were producing high amount of energy, ATP. This was very good. And the bad thing was that once oxygen developed, and such cells or microbes, when they were exposed to the oxygen, reactive oxygen species, dangerous molecules were made, and if this organism have to survive in the aerobic environment, then they must develop defenses against reactive oxygen species, which I will mention later in detail. Those are dismutases, catalases, and peroxidases. So first let me discuss about the differences in these mechanisms. Let me tell you first of all, aerobic respiration. Aerobic respiration is the most modern cellular respiration. It's the most evolved mechanism. It's the most efficient mechanism, right? And all of you know it very well, but I will just review it very briefly. That let's suppose if you are going to use the aerobic metabolism and you are going to utilize glucose as a substrate to make energy, then you know that through the process of glycolysis, glucose is broken down into pyruvate. And this process where glucose is broken down into pyruvate, this process is called glycolysis. Then you know that once pyruvate is made, then it is taken into mitochondria, right? And there are Krebs cycle going on. I will just make Krebs cycle here, right? It's a very simple diagram. This is Krebs cycle and pyruvate will enter into it, right? And it will pass through Krebs cycle, which is also called, right? So this is Krebs cycle. Now after that, electrons which are released from the breakdown of glucose and its breakdown products, they are taken to what is this? Electron transport chain, and eventually they will be going to the final electron acceptor, right? And in aerobic metabolism, final electron acceptor is, yes, this is oxygen, molecular oxygen, molecular oxygen. So what we say that aerobic cellular aerobic respiration is the most developed, most modern, most efficient, right? Why? Because it produces maximum amount of ATP. It consists of glycolysis, then Krebs cycle, then electron transport chain, Krebs cycle, and then electron transport chain. And while we are breaking down these products, finally these products will be glucose and its derivative products will be eventually broken down into carbon dioxide and water, and lot of energy will be produced, and that energy will be trapped in the form of ATP. But when these glucose and its breakdown products are breaking down progressively, they are releasing electrons, and these electrons will pass through electron transport chain, and eventually these electrons will be accepted by molecular oxygen. These electrons will be accepted by molecular oxygen, and this oxygen then will react with protons and make water, with hydrogen, react with hydrogen and make water, and energy will be trapped. I will not go into detail of the pathway. Now, this is fully developed structure. How it compares with anaerobic fermentation and anaerobic respiration, that is aerobic respiration. Now, the most ancient pathway, primitive pathway to produce energy by the microbe was anaerobic fermentation. This is the most primitive and most, I must say, rudimentary metabolic pathway to produce ATP. Then there was little more advanced, that was still anaerobic pathway, right? And that is anaerobic respiration. Now, what is the difference between anaerobic fermentation and anaerobic respiration? Let me explain. In anaerobic fermentation, if glucose is taken, right, glucose will pass through, it is passing through glycolysis and making pyruvate, pyruvic acid. Now, this glycolysis pathway is common in all of them. Again, here is also glucose and going through glycolysis pathway and eventually producing pyruvate. Now, this pathway, right, this is common in all three of them. It means it is present in anaerobic fermentation process. Glycolysis is also present in anaerobic respiration, cellular respiration, and it is also present in aerobic cellular respiration. Now, some differences will occur. Actually, in case of anaerobic fermentation, there is no Krebs cycle. There is no Krebs cycle. There is no electron transport chain, and as a final electron acceptor, oxygen is not used, right? Is not used. No Krebs cycle, no electron transport chain, right? And not utilization of oxygen as a final. So what happens in anaerobic fermentation? That nutrients are partially broken down, right? And this last product of glycolysis undergoes some other reaction, and it can be converted, let's suppose it can be converted into lactic acid, lactate, or it can be converted into ethanol and carbon dioxide. And what is the final electron acceptor here? It is not oxygen. That is why it is called anaerobic pathway, because those metabolic energy pathways which do not use oxygen as the final electron acceptor, they are said anaerobic pathway. Now, in anaerobic fermentation, the final final electron acceptor are organic compounds, and these compounds can be pyruvate, or they can be acetaldehyde. Now, these are the final, what is this? Electron acceptor, right? So electrons come over here. Now, we move to anaerobic. This produces very little ATP. Why it produces so little ATP? Because nutrient is only partially broken down. It is not completely broken down, right? Now, we move to another anaerobic mechanism of producing energy. This mechanism is called anaerobic respiration. Now, what I'm going to do, I'm going to tell you the difference between anaerobic fermentation and anaerobic respiration. In anaerobic respiration, actually, their glycolysis is there, and also what is this? Krebs cycle is also there. Even electron transporter chain is also there. Electron transport chain is also there. So, but there is a big change. The final electron acceptor is not oxygen. That is why it is called anaerobic respiration, not aerobic cellular respiration. Here, the final electron acceptor is, yes, not oxygen. These are usually nitrates or sulfates or elemental sulfur or fumarate. If you cannot remember them, no problem. At least remember that in anaerobic respiration, the final electron acceptor is not oxygen. In anaerobic fermentation, final electron acceptor is not oxygen, because in these two conditions, oxygen is not the final electron acceptor. That is why they are called anaerobic. But they are even having difference from each other. Out of them, anaerobic fermentation is most primitive. Anaerobic respiration is slightly more evolved, and aerobic respiration is most evolved, most advanced. This has only glycolysis, right? Fermentation has only, anaerobic fermentation has only glycolysis, no Krebs cycle, no electron transport chain, no utilization of oxygen at the end. Is that right? It produces electron acceptor pyruvate or acetaldehyde. Because there is partial breakdown, so there is very little ATP made, right? Then later on, bacteria evolved more, right? A more advanced anaerobic mechanism was anaerobic respiration. Now here, glycolysis mechanism is there, Krebs cycle is there, electron transport chain is there, right? So it is much evolved, much better evolved as compared to fermentation, right? But anaerobic fermentation, but still it is considered anaerobic, even though it has glycolysis, as Krebs cycle, electron transport chain, still it is considered anaerobic mechanism because it is not using oxygen as a terminal electron acceptor. It is using some other inorganic or sometimes organic compound, inorganic like nitrate, sulfate, or elemental sulfur or fumarate, right? But then is the most advanced mechanism. What is that? Aerobic respiration. Aerobic cellular respiration has glycolysis pathway as our cells. Glycolysis pathway, they are having Krebs cycle, they have electron transport chain, and in the end, as final electron acceptor, we use oxygen, and this helped to produce a lot of energy, right? So now we will see some of the bacteria will have only energy producing only by anaerobic mechanisms, right? And we will talk about later, there are some bacteria which have evolved further and they are having anaerobic mechanisms still with them, and also they are having aerobic mechanism of energy production. Now, if a bacteria has energy production only by anaerobic mechanism and bacteria cannot use oxygen for energy purpose, it means such bacteria which are having only anaerobic mechanisms, like they can do anaerobic fermentation or anaerobic respiration, such bacteria will only able to survive and multiply in anaerobic environment. For example, medically, such bacteria are present in big number between the teeth and gingival crevices. There is anaerobic environment, right? Then anaerobic are also present in colon, large intestine, right? Because those bacteria know how to survive without oxygen, right? Those bacteria know that they can produce energy in an anaerobic fashion. Is that right? Then there is another group of bacteria, we'll talk later. They are really very, very sharp. You know what they are doing? They have evolved fully to develop the aerobic respiratory pathway. But the clever thing is they still maintain anaerobic metabolic energy pathways. So if this is a bacteria which is maintaining its primitive metabolic pathway for energy, like anaerobic fermentation as well as anaerobic respiration, and it is evolved so much that it has also evolved the biological mechanism, right, for aerobic cellular respiration. This is a very lucky bacteria, because if oxygen is not present, still it can survive well. It can produce enough energy at least by anaerobic mechanisms, and if there is oxygen available, then it will multiply more efficiently in the presence of oxygen, as oxygen is providing at the end with more ATP. But if let's suppose there is another bacteria, this is very primitive bacteria in its biochemical reactions, that it can only do anaerobic fermentation and anaerobic respiration, right? But it cannot use the molecular oxygen as final electron acceptor. Then this bacteria will survive well in anaerobic environment and multiply well in anaerobic environment, but this bacteria cannot take extra advantage of oxygen. If oxygen is present, it cannot take the advantage of oxygen to utilize oxygen as a more efficient energy producing mechanism, right? So this was one thing, right? The relationship of metabolism, energy metabolism of bacteria and the relationship with oxygen, right? Then second relationship with oxygen, that is extremely important. As I mentioned in the beginning of the lecture, that when microbes are exposed, it does not matter microbe is having what energy metabolism, once they are exposed to oxygen, right, they will make lot of reactive oxygen species, which I will explain that how it is. After the break I will explain how reactive oxygen species are made. And reactive oxygen species are such atoms, molecules or radicals which are oxygen derived, but they are very, very reactive. They will react with DNA, they will react with proteins, they will react with your lipids, right? So they are able to destroy nucleic acids, DNA and RNA. They can destroy proteins, they can destroy lipids, and of course, if they keep on doing these things, they destroy the cell or microbe. So, as more and more oxygen was available in the atmosphere, right, bacteria had to evolve the mechanism to develop a defense against reactive oxygen species. Bacteria were forced to develop defenses against reactive oxygen species. Let's have a break, and after that we'll deal with four things. Number one, in the presence of oxygen, how reactive oxygen species are made. Number one. Number two, why they are so dangerous. Number three, that how bacteria, more evolved bacteria, have developed defenses against the reactive oxygen species like enzymes, dismutases, catalases, peroxidases, right? And I will also discuss those bacteria which fail to develop these defenses against reactive oxygen species, what kind of troubles those bacteria have. Let's have a break now.